Genetically modified organisms are becoming more common in our everyday life, ranging from designer dogs, stronger tomatoes, and cloned sheep. In all our scientific discoveries, though, very little effort has been put towards the longevity and better protection against disease in livestock. Mastitis is the most common cause of dairy cow deaths, costing farmers millions of dollars annually. This little bacterium has the ability to destroy farmers' livelihoods until it comes face to face with lysostaphin. Welcome to the genetic basis of stuff and things. First diagnosed in 1917, bovine mastitis is predominantly caused by Staphylococcus aureus, a fatal mammary gland or udder infection causing inflammation and diseased milk production. Cows can contract mastitis during any stage of their lactating cycle, particularly when an animal injures its udder, exposing it to bacteria. The infection causes extreme discomfort, and bloody discharge accompanies any milk produced. Ultimately, milk yield is severely affected, and animals suffer weight loss and dehydration. The most common way of diagnosing mastitis is the California mastitis test, which can help to provide early detection and prevention of disease. It works by mixing 4 small samples of milk with a California mastitis test reagent, which can detect the presence of excess somatic cells. Milk that reacts strongly with the reagent indicates that the cow has a mastitis infection. Currently, there has been no effective vaccine invented, but today's current cattle treatments of mastitis include intramammary antibiotics, which only work when a quarter of the udder is infected, or by culling infected animals to prevent further spread throughout a farmer's herd. Both of these options are flawed. If a cow's infection is not identified quick enough, then it can quickly spread from the individual mammary gland in one quarter of an udder into another, and even if administered only has a 15% cure rate. Also, due to the nature of the treatment, any milk that the cow produces within 5 to 7 days post-treatment must be discarded as not to contaminate the rest of the milk supply. There is also concern that the continued use of antibiotics in agriculture will give rise to drug-resistant bacteria. In 2005, another option was brought to light, when Dr. Robert Wall and his team developed the first transgenic cows with the ability to resist S. aureus. He did this by breeding cows that produced lysostaphin in their milk, a particular type of bacteriocin with the ability to ward off mastitis. Bacteriocins are peptides or proteins produced by bacteria, typically as an immune response or survival mechanism. Lysostaphin is an endopeptidase which is able to cleave the glycyl-glycine bonds found in the cell walls of gram-positive S. aureus, lowering infection rate and preventing recombinant production. Dr. Wall first experimented on mice, genetically engineering them to produce lysostaphin proteins in their milk. He came to conclude that transgenic production of lysostaphin by the lactating mammary gland will confer substantial resistance to Staphylococcus mastitis. Ultimately aiming his research to the prevention of mastitis in cows, Dr. Wall transferred what would become nuclear donor fibroblasts from 6 calves. After embryo culture, 650 blastocysts containing the transgene for lysostaphin were transferred into 330 recipient heifers. This resulted in 8 transgenic calves being born, with 5 surviving into adulthood. The lysostaphin levels in transgenic calves' milk was compared with non-transgenic calves and bacterially derived lysostaphin, and was found to be around 15% as active. This led Dr. Wall to conclude that mastitis resistance had a low heritability rate without the aid of selective breeding, and that lysostaphin produced in transgenic cows' milk was at a much lower rate than what was previously recorded in similar experiments done on mice, goats, and sheep. This could potentially be due to the components in cow's milk inhibiting some of the expression of lysostaphin. The animals received intramammary injections of S. aureus, and transgenic cows with lysostaphin concentrations of 0.9 to 14 milligrams per milliliter were able to ward off mastitis. Cows with the lowest S. aureus lytic activity had higher infection rates, and those with the highest lysostaphin levels were completely protected against infection. The cows that did develop any infection also experienced lesser symptoms. Increased levels of lysostaphin in milk and whether it has an impact on food safety is still an area for future study, but some research has found that it has little effect on the production of yogurt and cheese, although it's still present after the milk has been pasteurized. This is the process by which milk is heated to 63 degrees Celsius for 30 minutes to kill any harmful bacteria, including S. aureus, and make safe for human consumption. S. aureus can also infect humans, causing mastitis infections in breastfeeding mothers. Antibiotics have long since been the best course of treatment, but due to the increase of drug-resistant bacteria, other options such as lysostaphin are needed to be considered not only in the treatment of bovine mastitis, but also in humans. Current research has found that lysostaphin can have therapeutic benefits against drug-resistant S. aureus, although it still needs further refinement as it has too short of a half-life to make a major impact on an active infection and can also cause an autoimmune response. Dr. Wall and his team were one of the first to try and improve animal well-being and health through genetic engineering, and although further study in this area is still being done today, what has already been discovered has been a huge step for the dairy industry. Hopefully one day, lysostaphin and transgenic cows will be able to make mastitis a thing of the past.